The overall goal of this procedure is to enable non-invasive, economic, and efficient sexing and marking of altricial chicks as early as possible after hatching. This is accomplished by first taking buccal samples from newly hatched chicks. The second step is to trim the down feathers in a specific pattern, which stays recognizable until 10 days after hatching. Next, the DNA is extracted from the buccal samples using the Kelex DNA extraction method. The final step is to perform PCR using primers that amplify a sexually dimorphic region on the Z and W chromosome of birds, and load the samples on a gel to visualize the band pattern. Ultimately, on day 10 after hatching, each chick in a nest is identified by its unique haircut and a leg band is applied.
The main advantage of this technique over existing ones is that it is non-invasive and much faster than sexing from blood samples using conventional DNA extraction methods.
In general, people new to this method will struggle because they are not thorough enough in taking the buccal swab, which is key for a successful result.
To begin, prepare 50 milliliters of 5% Kelex 100 solution in molecular grade water. Constantly rehomogenize the suspension with a magnetic stirrer to prevent the Kelex resin from precipitating, while aliquoting 200 microliters of the solution in standard 1.5 milliliter reaction tubes.